Once upon a time, scientists in 1975 noticed that when mice were injected with endotoxin, their tumor started shrinking. This is when Carswell et al. First spotted TNF in action. In the same year, Kohler and Milstein developed hybridoma technology, fusing immortal myeloma cells with B lymphocytes to produce monoclonal antibodies, identical antibodies that target a single protein. In the 1980s, doctors found elevated TNF levels in diseases such as rheumatoid arthritis and Crohn's disease, suggesting that TNF is also involved in inflammation. Two scientists from London, Maney and Feldman, showed that TNF is a central regulator of inflammation in RA. Their work suggested that blocking TNF could suppress the broader inflammatory response. Chronic inflammation is caused when there is excess activation of TNF alpha. When immune cells are activated, TNF alpha is produced. Initially, TNF alpha is in a transmembrane form which gets cleaved by the enzyme TACE, which converts it to its soluble form. Soluble TNF alpha primarily binds to the receptor TNFR1, which alters its conformation to activate it. TNFR1 activation leads to distinct cellular responses through formation of different signaling complexes. It can lead to either cell survival and inflammation, apoptosis, or necroptosis. For instance, the formation of complex 1 can lead to cell survival and inflammation. Binding of TNF alpha to TNFR1 recruits complex 1 proteins. This leads to recruitment of TAC1, which activates the IKK complex, which phosphorylates I-kappa B alpha, releasing NF-kappa B. Free NF-kappa B enters the nucleus, which triggers transcription of genes that drive inflammation. Also, TAC1 can activate MAPK, another key protein involved in the inflammatory response. When NF-kappa B and MAPK signaling pathways are inhibited, complex 2 forms, which is either RIPK1 dependent or independent. TRAD binds TNFR1, which then recruits SAD, activating caspase 8 and the caspase cascade, leading to apoptosis. Complex 2C, or the necrosome complex, induces necroptosis and inflammation. It's formed by RIPK1 and RIPK3. RIPK3 phosphorylates MLKL, triggering its oligomerization. Activated MLKL will translocate to the cell membrane and induce cell lysis. This will release DAMPs into the extracellular environment, driving inflammation. Chronic inflammation occurs when there is excess TNF alpha. So wouldn't it be great to be able to inhibit TNF alpha actions through targeted therapy? In 1993, two scientists at Centocor, Vilcek and Knight, set out to combat TNF alpha using monoclonal antibodies. They created CA2, later termed infliximab, the antibody in Remicade. It's a recombinant chimeric IgG monoclonal antibody, part human, part mouse. Infliximab binds to soluble and transmembrane TNF alpha, blocking receptor interaction and downstream signaling. This lowers cytokine, chemokine, and adhesion molecule activity, while also inducing apoptosis in T cells and monocytes via a cascade-dependent pathway and mitochondrial release of cytochrome C. These reduce inflammation and make it effective in autoimmune diseases such as RA and IBD. Autoimmune disorders occur when the immune system mistakenly attacks healthy tissues. IBD includes Crohn's disease and ulcerative colitis, where the immune system targets the digestive tract. Immune cells release TNF alpha, triggering inflammation. Common IBD symptoms include the following. Similarly, rheumatoid arthritis causes synovial inflammation from excess TNF alpha, leading to synovial thickening, cartilage loss, and bone erosion. Common RA symptoms include the following. In Crohn's disease, a phase 2 trial gave a single infusion dose of 5, 10, or 20 milligrams per kilogram of infliximab to 108 steroid-refractory patients. At 4 weeks, 65% responded versus 17% on placebo, and 33% achieved remission, marking the first biologic to modify disease and earning FDA approval. The phase 3 Accent trial of 573 patients showed that every 8-week dosing for CD maintained remission in 45% of patients, twice the rate of the placebo. In RA, early trials of infliximab in 20 severe patients showed symptom improvement with 4 or 10 milligrams per kilogram doses. The phase 3 Attract trial confirmed this. Over 400 patients on methotrexate were randomized to infliximab at 3 or 10 milligrams per kilogram every 4 or 8 weeks, or placebo. By week 54, approximately 50% of patients on infliximab showed improvement in RA symptoms compared to just 20% in the placebo group. This led to FDA approval. These trials proved that blocking TNF alpha could transform inflammatory disease care. Remicade later had label expansions for many other autoimmune disorders. The clinical trials showed that infliximab was well tolerated, with side effects being mild to moderate. Although Remicade is effective for autoimmune diseases, it has limitations. IV infusions require frequent hospital visits, but longer-acting formulations or new subcutaneous formulations which let patients self-inject at home may improve convenience. About 20% of patients don't respond, and 20 to 30% develop antibodies against infliximab, sometimes needing surgery. Combination therapy with immunosuppressants like azathioprine can reduce resistance and improve effectiveness. Side effects remain a concern, so reducing them is an important goal. Remicade, the first monoclonal antibody, proved that targeting a single cytokine could transform autoimmune diseases, paving the way for future biologics and improving the lives of millions.